Yes, so my name is Katrina Exter. I'm from VLIS, the Flanders Marine Institute, and I'm going to be talking today about the data management that we are doing for EMO BON. But first, a little bit of a digression, a bit of philosophy. So data is the rock on which science rests. So observations of the world around us lead to hypotheses, and these are then improved or disproved by further observations. So without these observations, without this data, there can be no science, no discovery, no knowledge, no understanding. So that makes data absolutely fundamental, and the management of that data fundamental. So that makes data managers the masters of the universe. We hold the key to life, the universe, and everything. And that is us here all wandering about, and it's our job as data managers to shepherd all of those little individuals collecting their data. Now, science doesn't work in isolation. It never did. The first experiments would have been learning how to sort of put materials together to make homes or to make clothing, how to process food to make something yummy for dinner. Sharing that knowledge would have allowed early peoples to thrive. So now if we skip forward some thousands or tens of thousands of years, our current challenge in science is that we've got so much of it. We've got so many sort of sub topics that can be studied with new techniques, new instruments, and huge amounts of data that are being created all of the time. And in addition to that, science is carried out all over the world, and it benefits from that. We do, and we continue to, and we should continue to work together, not only in the same lab, but with anybody, anywhere in the world, by sharing our data with each other so we can co-create our science. Now, it's not enough just to share the final results and the conclusions with everybody else. We have to share the data, the foundational observations on which our science is drawn. So why is this important? Well, first of all, it's not enough just to say, trust me, I'm a scientist. Right? We need to humbly provide the evidence for our scientific conclusions, and this provides trustability in our science. Next, it's because very little data has only one way that it can be used. You never know what other science someone else can do with your data, in what ways they can combine your data with theirs to come up with new findings. No one can possibly carry out all the experiments to study effects happening over large areas of the planet, over multiple timescales, over multiple ecosystems. Your data can help others to do their science. Thirdly, it's because no one study ever finds out everything, right? Each of us study a little bit here and a little bit there. And when we put these little bits together to get the bigger picture, we create a science that is more reliable, it's better, it's wider ranging, and it's more accurate. And so the data that's collected in these experiments have to be made available so that they can be combined. And last but not least, it's because where our experiments are funded by the public, by public money, then those data do not belong to us. The science belongs to us. The science belongs to the person who comes up with it. But the data belong to the public because the public paid for it. Now, open science and fair data are catchphrases in all EU funding calls these days, but it's more than that. Really, data are not ours, they belong to everybody. Now, I'm a data manager. I've been working at the open science team of VLIS for 7 years now, I think. So on the one hand, data managers help the scientists. They are the ones who are producing the data, and we help them to effectively and hopefully efficiently share those data. And on the other hand, we work with data technologists, IT specialists, to develop and promote the systems and the standards and the methods by which data can be managed and shared. Now, in these 7 odd years that I've been working in data management, there's one really big lesson I've learned, and I'm going to pass it on to you for free. It's not the technology that is a limiting factor to effective data sharing, right? We have loads of cool tools and technologies that can help us do this. The limiting factor are the humans. So data management is very much also human management. We, data managers, need to help the humans, the scientists, to understand, first of all, that the data need to be managed so that they can be made findable, accessible, interoperable, and reusable, and then help them to do that. And secondly, we need to change thinking about the importance of data and its relationship to your science. The endpoint of data management, and in fact, the endpoint of research, is not when the science paper is published. It's when the data are also published. They are shared via an accessible data repository. Now, there are many agents that are responsible for making data fair and open. It's not something that will fall on only one person. So, of course, first you have the scientists who collect the data, and they must do so with an eye to the eventual findability, interoperability, and reusability of those data. But the institute that employs the scientists, they must have open access focused policies, and they must support via processes and tools the scientists who need to do the data management. And the funding agencies should also specifically include funding for scientists to do data management as part of their research. The data repositories that should collect rich and standardized metadata, they should collect interoperable data, and they should provide the tools to help the data providers to provide data that are interoperable. And finally, oops, and that didn't work very well, but never mind. And finally, the journals that publish the science should also require the data on which the articles are based are shared via trusted repositories before the papers can be accepted. Okay, so it's not something that only one person has to do. A lot of people have to work together to make sure the fair and open data can exist. Okay, so in these last few minutes, I've been trying to explain why data management is sexy and important. We do work that really makes a difference because we help the scientists to manage and share their science. So it's not lost, so it's not stuck in a silo somewhere. So on the difficult days when I've had to deal with a swamp of data and a swamp of humans, I can think about this bigger picture, and the world looks all pretty again. Okay, so that's enough on the philosophy, and now we get down to the more useful and interesting part of my presentation. So I'm going to talk about the data management that we've been doing here at Liz, and we're still doing for EMBRC's EMO BON project. Now, first, a little introduction to EMO BON. EMO BON stands for the European Marine Omics Biodiversity Observation Network. And it's a network from EMBRC, the European Marine Biological Resource Centre. We have something of the order of, I think, about 26 stations, which are distributed, as you can see here, over the European coastline. We've been collecting samples since 2021, from the water column, from the sediment, and the hard bottom, which is basically the hard bottom of the ocean, via things called settlement plates. And we make collections, sampling activities every 2 months to a year. The samples are processed with standardized and open access protocols. They've been published on ocean best practices. The samples are then sequenced twice a year, and all the data are archived on the European Nucleotide Archive and are open access after 6 months of being produced. We also run a bioinformatics pipeline to produce species lists and functional annotations from these sequences. The data we deal with in EMO BON are diverse. We have the sampling and event log sheets, which include observations and measurements. We do have some images, specifically from the HARP substrates, images of these collection plates. We produce sequences, and from the sequences, we produce sequence lists and other bioinformatics outputs. Now, all this data is centralized on GitHub, and I will talk about the management we do on GitHub in a bit later. First, I'm going to tell you about how we've gone about making each of these data sets interoperable and reusable. Now, this was important to do from the very point of us receiving the data, because as the expression goes, shit in, shit out. It was really important that we get good data coming in so we can produce good data going out. Okay. So the log sheets, these are created by each station. We use Google Sheets for this. In order to make these log sheets interoperable and reusable, we spent a lot of time creating a standardized template for all of the data to be written into the log sheets. So we have fixed columns, fixed data types, and so on. Now, particularly important in this process, which did take a couple of months to come to, was that we worked together with the station sampling teams to come to our final templates, but that it was managed by one person, who is the EMO BON manager. You need to work with everybody because you need agreement from everybody so they feel a buy-in to that part of the project. But you need one person to manage it all because otherwise you're just herding cats and you'll get nowhere. All of the measurements on our log sheet, so any value essentially on the log sheet, every single column on the log sheet is linked to a control vocabulary. This way, everybody knows what each column in each log sheet contains. So that's every person looking at them, and it's every developer who wants to read those log sheets with code. Making the log sheets interoperable from the very beginning makes the management and use of the log sheet data later much more efficient. As we had a lot of terms in our log sheets that were unique to us, we created our own vocabulary, or our own ontology, in fact, to accommodate these. We published that on our GitHub site. And for those who are interested, this is a quick screenshot of 3 of the ontologies that we have, an example of how we define the term tidal stage and storage temperature. So back to the log sheets. Now, each row on the log sheet is for one collected sample of water, sediment, or the hard bottom. Each sample, each row, has a unique and permanent material sample identifier, a PID, which after a lot of trial and error, we constructed with the pattern shown here, EMO BON _ station name _ sample type _ iterator. Now, exactly how you construct the material sample ID doesn't matter, but you do have to make sure that it is a unique ID, and it's something that will not need to change throughout the course of the project. For us, the ID is very important because it's used to link the samples to the events, the observations to the events, the sequences to the samples to the events, the images to the samples to the events, and the species names to the sequences to the samples to the events. So it's one of the key identifiers that's linking all our data together. So it was very important to think carefully about how we went about constructing this ID. Okay, so next, the images. Now I have to say that, in fact, we don't really do much with the images at the moment. And I know most of you here are going to be interested in image data management, and we don't do an awful lot of image data management just yet. But the next presentation by Stefan will cover that. We do have a lot of images. I think at the moment we have something of the order of 1,000 of images. The scientists seem to love taking photographs of their settlement plates. But they're individually very simple images. Since each camera has its own way of naming the images as you take them off the camera, we allow the stations to keep these names. They not have to spend a lot of effort renaming them. But when they upload the images to the data management platform that we're currently using for this part of the EMO BON project, we require them to fill out, along with the images, a templated spreadsheet, which for each image lists the file name of the image, the event identifier, and various technical metadata that are related to that particular image. These files are then used to populate a standardized and interoperable metadata file, a CSV file, that we use to share the images and their metadata with the general public. Now, our intention is to eventually put these images and these metadata on a community repository, such as Bioimage Archive, but we haven't had the time to do that just yet. And there is an important lesson, a second important lesson that I give you for free. The reason we haven't managed our images properly or completely yet is because we didn't reckon how much human resources, basically how much of my time, was necessary to do that. The time to collect, gather, combine, standardize, and quality control the images and the metadata is something we hadn't budgeted for. We do have the processes to do it, essentially the same processes we use for the log sheets, but we haven't really got the time to do it. So never, ever underestimate how much time it can take to do data management, especially for larger projects. always budget for it. And even if you're writing a grant proposal and it doesn't ask for anything about data management, include it anyway, right? Data cannot be made fair, cannot be made open unless someone does the data management, and that requires a person and their salary. And this is by far, sort of human resources are by far one of the biggest upfront euro costs of our project. So the next data we have are sequences. We create several different types of sequences from our samples. We archive these all on the European Nucleotide Archive. Genoscope, which does the sequencing itself, they upload the data to ENA and we upload the metadata to ENA. So our data management process has to sort of coordinate the different agents doing the different activities. We use the material sample IDs that I mentioned before to link the sequences and their metadata to the sample log sheets on which we have various other information which we cannot put into ENA. So this links the data in ENA to the data, our data in GitHub. We provided Genoscope with a template spreadsheet for them to provide us back with various technical metadata regarding the sequencing process to provide provenance information about the sequencing. And we'll be linking these terms to Mixus terms and including them in our EMO BON ontology as well. The upload to ENA we do via their APIs, the metadata and data upload, to get the so-called accession numbers we do via their APIs. And this is quite easy to do because all of the metadata that the API needs is in our log sheets, which are interoperable already. So the process is a lot faster and a lot less error prone than it would have been either if you did it manually or if you didn't already have interoperable information. And then finally, we have data from the bioinformatics pipeline that we have to manage. The bioinformatics pipeline produces a lot of data, intermediate data, quality control data, final data, all sorts of files all over the place. And it's important for us to provide all of these to the public so that they, first of all, can trust our results, and secondly, so that they can reuse our results. So each output file produced by the bioinformatics pipeline needs to be described, what it contains, what each part of the workflow is that produced it, how you go about recreating it. This is done via a standardized metadata file, a row crate file, which I will explain in a few slides on. Now this metadata file, as well as describing the output files, we also need to describe the input files, right? That is part of the provenance. So the settings of the pipeline when it was run, information about the pipeline itself, and a reference to the sequence that was processed in that particular run. So by providing this who, what, where, when, and how of the bioinformatics data, we're providing the provenance information that will make these data reusable. Finally, for the bioinformatics, we also need to explain in a more granular detail those particular files that contain the final scientific results of the processing, being the species lists and the functional annotations, as well as the process sequences, the so-called FASTA files themselves. Now, each of these output files, these species lists, the FASTA files, the functional annotations, they contain information that needs to be understood without having to ask the bioinformatician what column contains what information, right? So these files need to be self-explanatory. Now, while it's true within the bioinformatics world that these particular types of outputs from almost all bioinformatics pipelines, they're structured in very similar ways, right? There is a sort of standard that is used. But they're not actually standardized to the point where you can understand the output fully from one pipeline and from another pipeline equally well. Okay. There are some things that are different. The IDs that are used in the rows, the way the columns are entitled, the way the taxonomic information is given, the type of taxonomic information that is given. For our outputs to be interoperable, not only to us, but to anybody else, we needed to create metadata files with descriptions of the data in each of these files, and that made them interoperable. Now, luckily for us, the bioinformaticians are part of the EMO BON project, and so we work with them together so they could modify their code to produce these outputs and produce these extra metadata files that made their outputs more interoperable. So we didn't have to do this post hoc, which saved an awful lot on time and human resources. Okay, so that's the individual different types of files, different types of data, rather, that we made interoperable and reusable. And so now about the data management. Now, EMO BON's data comes from and goes to several different places, and we have several different types of data providers in the project. So we have the marine stations and their log sheets, a Genoscope and its sequences, and we have the bioinformatics team as well as the management team. These groups have got different needs, they have different backgrounds, they produce data of different formats and degrees of interoperability and so on. So we chose GitHub to centralize our EMO BON data collection, our EMO BON data management. And this is just a screenshot of our GitHub space. So all data are uploaded to GitHub on regular timescales, and after upload, that triggers a quality control process that's specific to each type of data file. They're supplied with metadata, and they're made to make them interoperable. And once you've created a workflow that does this for one particular data type, then it happens from then on more or less automatically, and that speeds up the data management. We decided to allow each team of data providers, data producers within EMO BON, to work the way that's best with them, producing data that they are familiar and comfortable with. That's why we're using Google Sheets for the log sheets, for example. The work that we did in the data management was to ensure that these data were interoperable in the ways that I explained before. So formatting and standardizing the data. So we worked on formatting and standardizing the data collection forms, the Google Sheets. We worked with them to add provenance and descriptive metadata after we had uploaded them to GitHub. And in some cases, for example, for bioinformatics, working directly with the data creators to add interoperability directly to their workflows. Now, why do we choose GitHub? There are a number of reasons. GitHub has version control. It allows for different levels of access. It allows for automatic actions to be implemented. We can build mini web pages from it. You can raise issues on repositories to point out errors or request changes and so on. It's good for a project where the team is highly distributed, but where the management and provision of the data in one place is particularly important. We have workflows that can reformat our data into whatever is required when we want to publish those data on data portals and repositories. So ENA, we've already published our sequences on there, but we will be submitting data to Eurobis/eBonNet for the biodiversity results. We'll be sharing our data with Blue Cloud and so on. And we can reformat our data into whatever they require by making sure that our data are interoperable. It's easy enough to write code to do that. Now, one thing that GitHub doesn't allow us to do is to store large files. The free space you get is too small for that. And for our bioinformatics, this is a problem. So the solution we came up with is to use something called large file storage, LFS. The files themselves are stored somewhere else. So we have an EMBRC disk somewhere. But they're intimately linked to GitHub so that they appear to be on GitHub, and they can be managed on GitHub together with the data that actually are there. And then the data packaging. This is about how we package together our data into logical units. This helps with the data management and at the same time allows us to add technical, descriptive, and provenance metadata to the files in the packages. So we use something called RO-Create, Research Object Crate, to do this. RO-Create is a community effort to establish a lightweight approach to packaging research data with their metadata. The data file in each crate, each package, can be of any type, any location, any size, etc. One crate is everything that the crate owner thinks should be considered a unit. So there's no predefined way that a crate should be constructed. There's no predefined way that particular files need to be packaged together. It's entirely up to you how you package your data. So for our crates, for example, we've got for each observatory we have with the log sheets and their QC, we have one crate. The sequencing information is another crate. The bioinformatics outputs are other crates, and so on. Now, what is an RO crate? It's actually just a metadata file created following a standardized schema in a language called JSON-LD. This is a language that's machine understandable, machine readable, and machine understandable. It's text-based. It's actually also quite easy for a human to read, and it's recommended and used by the World Wide Web Consortium. So within these row crate metadata files, we have the who, what, where, when, why, and how of all of the data within that crate, provided in an interoperable way, and all the links between the data in that crate and data in other crates are also given. We find it to be a valuable data management tool, a valuable interoperability tool, where you have data that are not just sort of single files, but you have whole collections of files of different type and potentially different location, but they need to be treated individually, but they need to be managed together. Okay, and finally about our data landing page. So as I said, we will be sharing our data through the standard community repositories, ENA, EMBL.net, Eurobis, and so on. But we also have something called a data landing page. And this is our way of sharing data for the quote-unquote general public. Now, at first, we just wanted to provide a more comfortable navigation experience for people who are not familiar with or comfortable with working their way through GitHub. So we created this so-called data landing page to create a sort of website look to the GitHub space. But we soon discovered that this is also a good way to share data and metadata, making it more accessible, findable, and reusable, and that for both machines and for humans. So this is a screenshot of our data landing page, the top of it and a bit in the middle. Now, on this data landing page, a human can browse our various data packages, as you can see here. These are, in fact, our row crates. These are built from our row crates, and when you click on them, you are viewing the information in the row crates. They can find particularly quickly results that we want to highlight, for example, the latest release sequences or bioinformatics outputs. And we will be putting more on here. Our idea is to put a Jupyter notebook that will allow you to browse and to work on all the EMO BON data. So the data landing page provides better accessibility to the humans, but it also provides better accessibility to machines. Now, what machines would this be, you may ask? Data harvesters, for example, Blue Cloud or EOSC, Google and other search engines, or developers wanting to access, for example, updates to the data set that they downloaded previously. These are sort of three examples of machines. So we're doing this at the present in three ways. First, by embedding what's called fair signposting, which allows harvesters to developers to know how to access and get the data in the format that they want it, because we do provide data and metadata in multiple formats. By embedding search engine optimization to allow search engines to find the pages and all the data, and that is these data packages, the data files, and eventually also data points. And then finally, we have, or will be having rather, linked data event streams that will pump out metadata for machines to read on a regular basis so they can keep getting updates. And that is the end of my presentation. And I guess I stopped screen sharing, or do I stop screen sharing now?
I think Stefan can take over if you just...
Yeah. Okay, here we go. Voila.
There we go. Hello, everyone. I'll try to finish at 10:20, but I guess I'll go a bit further. Hi, everyone. My name is Stéphane Pesan. I'm a marine biologist. I work at EBI as bio-curator, and my role is mainly to link up with marine science projects with the community and link them with the different services and archives that we have at EBI. And today I will talk about some exemplar marine science expeditions that we've been working on at EBI for 15 years now. So a quick note about EBI, what EBI does. We help scientists realize the potential of big data in biology and offer services to exploit complex information. Of course, the scope of EBI is quite large. It covers, obviously, human science, health, and there is a growing body of environmental data being archived at EBI and being analyzed at EBI. That covers all biomes, but I would say that the marine biome is really a good exemplar. It's well developed. The marine science community is usually a good data managing community. It follows good practices. It usually develops quite good practices as well. So today I'll talk about a few of those exemplars. A first one that you might have heard about is Tara Oceans. That was 15 years ago. Tara Oceans looked at plankton, so the marine microbiome. The specificity was to collect samples for DNA, RNA, as well as imaging, and splitting that into different size fractions, looking at everything from viruses to small eukaryotes. And it had one research platform, Tara, several devices to sample, and several devices to analyze the samples as well. Another one is Atlantico Mission Microbiomes, which is more recent, 4 years ago. That one was also on board a single platform, Tara. It expanded the complexity of the data types, included, as in Tara Oceans, DNA, RNA, and imaging, but also proteomics and metabolomics. So adding to the complexity of the data we need to work with. And then there is TREC, which basically follows the same approach, multi-omics, but then added complexity in having several sampling platforms, again, Tara, but also platforms on land, and also extending the environments that we sample from the marine to the coast and moving inland. So adding new protocols, but also the need for new vocabularies to describe the different environments. Back to this image of multi-omics. At EBI, we have different resources, different archives, and analysis services that cope with these different types of data. So we have ENA, the European Nucleotides Archive, to work with DNA and RNA. We have the PRIDE platform to look at proteins and the proteome, the metabolites platform to look at the metabolome and describing metabolites. And we have the bioimage archive to archive and annotate images. On top of that, or maybe before that, we also have BioSamples, which is an archive and a curation platform as well for sample provenance and sample annotations. And that is common to all the archives. It's fully integrated with ENA, meaning that everything, all the samples at ENA are also described as BioSamples, and there's some synchronization. With PRIDE Metabolites and Bioimage Archive, there is to some extent that possibility. It's not fully integrated yet, but we encourage, at least in those projects, we use BioSamples for all the archives, and that allows the integration of the data across the different data types. And we were pushing to to make sure that this is adopted in most marine communities and beyond. So what I'll do today is not talk about data management of the different data types. I'll focus on the biosamples, on the provenance metadata, and the type of how important it is to describe them nicely. So why biosamples? What does it offer? Biosamples is it's European to start with, and it's hosted at EBI. It offers simple upload submission workflows, and it also has a help desk. It's not perfect, but it's always improving with new checklists. But I'll talk about that. It offers persistent identifiers, accession numbers. It offers, to some extent, metadata validation. We'll see that in the next slides. Validation can be quite time demanding or resource demanding. So there is always place for improvement and development, but at least for the minimum information, there is good validation. And then it offers discoverability of metadata across the platforms. The metadata from BioSamples is also progressively being discovered by the Blue Cloud, so that's a big plus. So molecular data is usually not so much integrated in the big data, at least for biodiversity. But now it's being integrated in Blue Cloud, OBIS, or GBIF, so we're getting there. And as I said, BioSamples fosters the integration across the multi-omics. So once we plan to submit metadata, provenance metadata to BioSamples, what helps a lot is using standards or checklists. There are several checklists that can be used at BioSamples. There are the very minimal ones. That's the first one in this list, which is the BioSamples default. ENA also has a default, which is expanded a bit on the BioSamples default. And then there's a series of MIXS from the Genomics Standard Consortium. So MIXS is minimum information about any sequence. So the MIXS has different checklists for the different environments. They usually, the four that I showed there for air, sediment, soil, water, which is the biomes that we use and that we sample and TREC, they share a fair bit of common metadata fields, but depending on the environment, they also have additional ones. Yes, all of them have mandatory fields, optional fields, and when the information is missing, we use this one, which is not yet an ontology, but it's at least a controlled vocabulary from INSDC, with controlled terms to describe when the information is missing. Useful ones, for example, missing when it's a control sample. At least we can use that controlled vocabulary, but there are a number of them. Then, if we look at the minimum checklists, the very minimum from BioSamples is taxonomy. Then if we look at the second and third ones, that's from the ENA minimal checklist. It has to have a geographic location, but not yet latitude, longitude. The minimum requirement is a name of the location. That's a very minimum, and a collection date. So that's bare minimum, not enough, in my opinion. Then if we look at one of the MIXS checklists, this one is the water checklist. Then you have, in addition, then geographic location, latitude, longitude, depth, and then three terms that describe the environment at broad scale, local scale, and the environmental medium. We'll look at that. And project name. These are mandatory from the Mixes Water checklist. Yes, now what I'll do is go through each of them with some explanation. So for taxonomy, at EBI, at ENA, we use the NCBI taxonomy, which is quite large. It has 3 million entries, but it is composed of a number of linear taxonomy, like this first example, where we provide in BioSamples the name, the scientific name, the source, and the unique ID in the form of a URL. But NCBI also includes non-linear taxonomy. So when you collect plankton on a filter, for example, it's a community. So still they have taxonomy to describe that community, because taxonomy is mandatory. So we need terms to describe those. A very common one in marine science is marine metagenome. But if you collect the microbiome on the skin of a fish, you would have fish skin metagenome as a taxonomy. So that's one way to describe the taxonomy, even though it's not linear. In marine science, people like a lot, and it's a good thing, to use worms as a taxonomy backbone. It's very good because it's well curated. So at EBI, we have this matching between the worms taxonomy and NCBI. So as I said, 3 million taxa at NCBI, 250,000 accepted marine species around that at worms, and we can match, and the match is done by worms actually. We can match 98,000, and usually in most data sets, 99% of the taxonomic, the scientific names are matched between worms and NCBI. So it's quite useful and easy to interoperate. Geographication, country and or sea. We use the controlled vocabulary from NCBI. So it's a text choice in terms of validation. And that list includes countries, of course, but also major oceans and world regions. So it's a first step. And it's if everybody does that, then it is useful. Then if we look at the collection date, geographic location, latitude, longitude, and depth, the validation is restricted text. So there's a regex check on those. Well, that's the regex below, but it means that we use the ISO, the corresponding ISO standards in the regex, and depth includes a number of different terms, but it's restricted. The units are meter or millimeter, which is an issue at the moment because we don't know exactly which unit people used. But it's, yeah, it needs lots of curation to make sense of that field at the moment. Yes, then the broad scale, environment, context, and the local and medium. These are free text. That can be improved quite a lot. So what we do is we recommend, when we do the curation, we use ontologies, but not everybody goes through a data manager. So we get lots of different free text values at BioSamples for these terms. But using ontologies makes life much easier when you try to integrate the data afterwards. What we use is the ENVO. So examples for the 3 levels, for example, estuarine biome for the broad scale, marine wind mixed layer or epipelagic or photic zone, these are all local, what we consider local environmental context terms. And seawater is a common one for the environmental medium. So using those, again, providing in BioSamples the name, the source, and the unique ID in the form of a URL. Yes, then we get into the optional ones. And yes, geographic location, region, locality. That's quite useful. And we use the marine regions vocabulary registry to provide terms there, things that are a bit more specific, like marine Atlantic Ocean or an Atlantis seamount, for example. If you sample in a specific feature, you can specify that in the geographic location. I'll speed up a bit. Then the checklists have different groups of attributes. Sample collection can be described. So the amount or size of sample collected. This one has a restricted text with regex. Biomass, organism count. That is unfortunately free text, but it's part of the checklist. Then attributes describing the method, storage, and transport. These are common ones that were added even from TerraOceans, I believe. So things like the sample collection device, the collection method, the size fraction, which is also, although it's controlled, validated with a regex, it needs a lot of curation because people tend to put anything or everything, including text. Sample storage location and temperature, storage temperature. So these are all things that can be captured and are actually part of the checklist. Then you can also include information about the local environmental conditions, so pressure, conductivity, temperature, fluorescence, and there are lots more environmental parameters included in the checklist for the different environments. And these usually have a regex as well, so there is some level of validation. But they are all optional, so which means that you can submit your sample metadata without that information, and the value of having a data manager in your projects or initiatives is that they can spend time on going into the optional fields and making the metadata much better. So back to the exemplar marine research programs. What we've done in these is to, yes, early on, we have unique identifiers that we use for the samples. We use them at the time of sampling. We identify the samples with a barcode on a sticker. And we use also a system of log sheets, with the difference that they are physical log sheets. Having digital log sheets is marvelous, but if you are in the field and if it's intensive, it's more difficult to capture everything digitally from the start. So in our projects, we tend to push for a first step as physical paper log sheets, where we can put the equivalent barcode on the log sheet, and the log sheet looks like this, and that's where we capture the required metadata for the provenance, but also, so this first sample, I hope you see my mouse, on the top left corner, that one, for example, would be a sample collected at 20 meters using protocol S320. Then you need in the metadata to convert that in more detailed information, but that S320 means it's a sequencing protocol. It's a size fraction 3 micron to 20 micron. You have information about the, it was stored in a cryovial, 5 ml at -80, and the filtration volume is mentioned and the filtration duration. So we put in as much as possible the basic metadata that is required. But that needs to be curated afterwards. The AI is helping us quite a lot now to extract all the information, either extracting the values themselves, the barcode values, but in terms of comments, when we have comments, then we sort of crop the region where there's a comment, and that is made available rather than trying to read to recognize the writing. 24. Yeah, I'm concluding. All that data can be submitted to BioSamples. I believe that will be covered in another webinar, so I'm not touching that, but it can be submitted through APIs. There's also a drag and drop, so the Google Sheets that you heard about in EMO BON, that could be converted slightly and then some uploaded, dragged and dropped in BioSamples to create the metadata. So the take-home message. First, take time to identify your data domains, the data types, and to describe the different protocols that you will use, and spend time describing those protocols. Then select rich metadata standards or checklists. We use Mixes, but there are others as well for other environments. Design the metadata collection and curation according to those standards, so that what you collect, the information that you collect on the log sheets, physical or digital, already match what the checklists are expecting. And that's basically that. Use standard formats, standard units to facilitate the validation, and use controlled vocabularies and ontologies even when the checklist is not asking for one. Do the effort because it helps in the long run. And that's it. I'm done. If you have questions, I think it's now the time.
Great. Thank you very much. Thanks, Katrina and Stefan, for sharing your knowledge, experience, and advice in data management with all our audience. And thanks to our audience for joining today, this webinar, and also asking your questions. So at the moment, I can take one open question, which is in there. But meanwhile, feel free to type in your questions as we go along, because we still have some time. So one question I think is probably for Stefan, for you. So for NCBI taxonomy, control vocabulary is considered more lenient. Is that the reason why it's adopted for marine microbiome?
There's probably a historical reason to start with, because for molecular data for DNA, RNA, there's this INSDC that I mentioned, which is collaboration between NCBI and GeneBank, so the DNA archive in the US, between them and EBI in Europe and the sequencing center in Japan, and they synchronize themselves. And NCBI has been the backbone for taxonomy for the molecular data domain, and that's why we use it as a base. But then having well-curated taxonomies for different environments, like worms for marine domain, that's a big plus. And having then matching the two is quite useful. But for example, worms historically has not looked at the microbes, the bacteria. It's not well described in worms, whereas it's very well described in NCBI. So they are complementary, and matching them as much as possible is a plus.
Yeah, I can add to that to say that I don't know if this was the original reason, but another reason why NCBI is good to use is because they use identifiers. And there are some taxonomies out there that don't use identifiers. And this is super, super important, because taxonomy changes every single second of every single day. So it's super important to have proper identifiers. Basically, it's an ontology, the taxonomy. Proper identifiers, so that when there are changes to a particular taxon, you can trace that through the use of the identifier and the way the identifiers are linked. New ones and the old ones will be linked to each other. And indeed, so NCBI and worms, as well as I imagine NCBI and other taxonomies, are working on cross-linking their databases so that you can get a worms ID for an NCBI one and vice versa, where that's possible. And that's work I think that's ongoing in a couple of EU projects. And that's very useful for making taxonomic results coming out from genomics more useful for the general biodiversity community as opposed to for omics special.